Hello students, today we are going to learn about chromosomal theory of inheritance. Okay, so before we go to learn about chromosomal theory of inheritance, we need to learn few more things. Okay, so we have already studied that Mendel is the person who was the very first person to perform experiments in genetics. Okay, so he performed his Mendel's laws of inheritance. He published his laws of inheritance, that is Mendel's laws of inheritance. Mendel's laws of inheritance, they were published in 1865 only. They were published in 1865 only, but they were not discovered until 1900. Okay, but they were published in 1865, but they were discovered in 1900. They were left unidentified. Mendel's laws of inheritance were left unidentified until 1900. Okay, until 1900 Mendel's works were left unidentified. There are some reasons behind Mendel's works left unidentified. That is because of the lack of communication in those days. Lack of communication in those days. Okay, it was in 1865, so there was a very poor communication systems in between the people. So there was a lack of communication is one of the reasons why Mendel's work was not identified. Okay, there was no publicity done based on this Mendel's work. No publicity was done. He just published it and there was no publicity because of lack of communication. So his work was left unidentified due to lack of communication. Okay, and the other reason is see, this Mendel has considered factors or the whatever the so called genes. Now we are calling these factors as genes. He said that these factors or genes they are going to undergo segregation. They are undergoing independent assortment. They are going to undergo independent assortment. That means there is no blending of the characters, no blending of the factors. He said that there is no blending of the factors, there is independent assortment of the genes, no blending of the characters is shown by the Mendel's factors or genes. Okay, but this is not true because people were seeing that variations are occurring in the living organisms, variations are occurring continuously, continuous changes are occurring in the living organisms. So it is clearly saying that there is blending of factors in the living organisms. There is a blending of factors, that is very clearly known. Okay, because there is variations occurring, living organisms are gradually changing. So this is thought to be false. Okay, not applicable in all the cases. Independent assortment, no blending of the factors, characters is not true in all the cases. Okay, then the next thing is Mendel, he was the first person to use mathematical applications for explaining biology. Okay, he used mathematical equations. He used mathematical equations. He was the first person to use mathematical equations in biology. Okay, he was the first person to use mathematical equations in biology, which was not understandable for many of the people of those times. Okay, and the other thing is that there was no physical proof. Okay, no physical proof. There was no physical proof for the existence of these factors. No physical proof for the existence of the factors. He couldn't give any physical proof for the existence of these factors. These are all the different reasons why Mendel's work was not identified until 1900s. Okay, lack of communication, Mendel's independent assortment, that is no blending of characters, was not accepted because there is very clear variations occurring in living organisms which is not acceptable, and mathematical equations were applied for the first time in biology, which was not understandable by many of the people, and no physical proof for existence of factors was given by Mendel. Due to all these reasons, Mendel's work was left unidentified. Okay, but later on there were some people. Let us see how this Mendel's work was rediscovered. in 1900 by 3 different people independently, like De Vries, Karl Correns and Schermark. Okay. These were the 3 people, De Vries, Karl Correns and Schermark. These were the 3 people who independently rediscovered Mendel's work. These 3 people were the ones who independently rediscovered Mendel's work, because by this time microscopic studies were advancing. Okay, because by this time microscopic studies were advancing. There were studies done on the nucleus. There were studies done on the structures of nucleus. Microscopic studies were advancing. Microscopic studies were advancing, and there was studies on the structure of the nucleus. There were studies done on the structure of the nucleus. Studies on the structure of nucleus were also done during that time. Chromosomes were identified. Chromosomes were identified. Okay. So, these are few points and we need to learn more. Okay, De Vries, Karl Correns and Schermark are the persons who rediscovered Mendel's works. Okay, these 3 people, they rediscovered Mendel's work, and because at that time microscopic studies were advancing, studies on the structure of the nucleus was done, chromosomes were identified. Okay. Okay, and then movement of the chromosomes, during meiosis, movement of chromosomes during meiosis was also studied. Movement of the chromosomes during meiosis was also studied. Okay, movement of chromosomes during meiosis was also studied during this time. Okay, so it was observed that the movement of chromosomes and the movement of genes are parallel to each other. Okay, movement of chromosomes and movement of the genes and movement of the chromosomes were parallel to each other. They were parallel to each other. Okay, it was also observed that movement of the genes and movement of the chromosomes are parallel to each other. Movement of genes and movements of chromosomes are parallel to each other. By using this concept, movement of chromosomes, Mendel's laws were explained. Okay, using movement of chromosomes, Mendel's laws were explained. Mendel's laws were explained. Okay, using this movement of chromosomes, Mendel's laws were explained. Okay, and during 1902, this chromosomal theory of inheritance was given. This chromosomal theory of inheritance was given by Sutton and Theodore Baveri. Okay, these 2 people have proposed this chromosomal theory of inheritance. Sutton and Theodore Baveri, they have proposed this chromosomal theory of inheritance. Okay, let us see what is this chromosomal theory of inheritance saying. Chromosomal theory of inheritance is saying that genes are present on the specific loci of the chromosome. Okay, this chromosomal theory of inheritance is saying that genes are present on the specific loci of the chromosomes. Okay, when these chromosomes are the ones which are undergoing segregation and independent assortment. Okay, so here genes are present on the specific loci of the chromosomes. Genes are present in the specific loci of the chromosomes, and chromosomes are the ones undergo segregation and independent assortment. Okay, so this is said by the chromosomal theory of inheritance. Chromosomal theory of inheritance is saying that the genes are present on the specific loci of the chromosomes, and the chromosomes are the ones which undergo segregation and independent assortment, not the factors. Because the genes are present on the chromosomes, they are also undergoing segregation and independent assortment, but actually the ones undergoing segregation and independent assortment are the chromosomes. Okay, let us now learn the salient features of the chromosomal theory of inheritance. Okay. Now we are going to learn the salient features of the chromosomal theory of inheritance. Salient features of the chromosomal theory of inheritance. Okay, so salient features of chromosomal theory of inheritance is saying that the links between one generation to the next generation is the sperm and ova. Sperm and ova, these are the ones. The sperm and the ova are the ones which are responsible for connecting the different generations. How a new generation is produced? From the sperm and ova only a new generation is produced. So, these are the ones which are helping in continuing the generations, continuation of one generation to another generation. Sperms and ova are responsible for the continuation of one generation after another generation, one generation after another generation. The sperm and ova, because they are connecting the generations, they must carry the hereditary characters. They must contain the hereditary, they must be containing hereditary characters. They must contain the hereditary characters. Okay, they must contain the hereditary characters. During fertilization, the nucleus of the sperm and ova fuse. Nucleus and this of the sperm and the ova, they will be fusing. During fertilization, the nucleus of the sperm and ova will fuse. During fertilization, nucleus of the sperm and ova will be fusing. Okay, so when nucleus of the sperm and ova are fusing and they are responsible for the new generation, that means inside the nucleus what are present? Nucleus is fusing. Because the nucleus is fusing, okay, the chromosomes must carry the hereditary characters. The chromosomes must carry the hereditary characters. Okay, because the nucleus is fusing, nucleus contains chromosomes, so the chromosomes must be carrying the hereditary characters. Okay, so the genes are present in the chromosomes. Genes are present in the chromosomes. Okay, genes are present in the chromosomes. Each chromosome has a specific function in the body. Each chromosome has a specific function in the body of the individual. Each chromosome will have a specific function in the body of the individual. Okay, and the loss of a part of a chromosome or the entire chromosome the loss of a part of the chromosome or the entire chromosome will result in the lack of the physical, it will result in physical or functional abnormalities. Okay, it will result in the physical or functional abnormalities. The loss of a part of the chromosome or the entire chromosome, it will result in the physical or functional abnormalities. Okay, and all the cells in the body are diploid. All the cells of the body are diploid. All the cells of the body are diploid. Okay, diploid means they contain two sets of chromosomes. They contain means they contain two sets of chromosomes, 2n. That means they contain two sets of chromosomes. Okay, except all the cells of the body has nothing but the somatic cells. Okay, somatic cells. Whereas the sperm and the ova, they are haploid. They contain only one set of chromosomes. They contain only one set of chromosomes. Okay, they contain only one set of chromosomes because the sperm and ova are haploid. They contain only one set of chromosomes. They are said to be pure. They are said to be pure because they have only one type of chromosomes, right? So that is why they are said to be pure. Okay, the gametes of sperm and ova are said to be pure. Okay, because they contain only one type of chromosomes, they are called as pure. Okay. So now during fertilization the sperm and ova will fuse, right? So sperm is haploid, ova is haploid. So these two will fuse during fertilization. It will give rise to 2n condition. Again the daughter individual is going to become diploid. In that way the sperm and ovum will help in maintenance of the constancy of the chromosomal number in the daughter individuals. Okay. So here the 2n condition is maintained again in the daughter individuals. Okay. Sperm and ova n both are haploid, fused together form 2n. Okay, this is the next feature of the next characteristic feature of chromosomal theory of inheritance. We have few more to learn, students. Next thing is these chromosomes do not undergo any blending. These chromosomes, they do not undergo any blending. Okay, that means the two chromosomes, they do not undergo any blending. There is no mixing up of genes between the two chromosomes. Okay, there is no blending, there is no mixing up of chromosomes between the chromosomes. So this chromosomes is considered as one unit each, and during inheritance also all the entire set of chromosomes are inherited together. Entire single set of chromosomes will be inherited together. So the chromosomes, there are no blending occurring between the chromosomes. The entire set of chromosomes will be inherited together. So that is why these chromosomes act as a unit. These chromosomes, they act as a unit. Okay, chromosomes, they act as a unit. Okay, and the next thing is the homozygosity. The homozygosity or heterozygosity. Dominance or recessiveness. Dominance or recessiveness. Okay, homozygosity or heterozygosity, dominance or recessiveness, whatever it is, it will be due to the type of chromosome and the behavior of the chromosome. It will be due to the type of the chromosome and the behavior of the chromosome. It will be due to the type and behavior of the chromosome. Okay, so it is going to depend on the type and behavior of the chromosome. Okay, students, so the homozygosity or heterozygosity, dominance or recessiveness, it will be dependent on the type of the chromosome and the behavior of the chromosome. Okay. So these are some of the salient features explained according to chromosomal theory of inheritance. Okay. So with this we finished to learn about chromosomal theory of inheritance. Now later on, there came a person who is called as T.H. Morgan. T.H. Morgan. Okay. So T.H. Morgan, this person has come after the salient features of this chromosomal theory of inheritance was given. This person T.H. Morgan has come, and this person, he on the basis of the features of chromosomal theory of inheritance, he tried to experimentally prove the chromosomal theory of inheritance. Okay, T.H. Morgan along with his colleague, he tried to experimentally prove the chromosomal theory of inheritance. For that reason, he has selected fruit fly. He has selected fruit fly whose scientific name is Drosophila melanogaster. Drosophila melanogaster. Okay, he has selected the fruit fly Drosophila melanogaster. There are some reasons for him to select Drosophila melanogaster. Let us see what are the features of Drosophila melanogaster. Okay, so this Drosophila melanogaster, it has a very short lifespan. It has very short lifespan. It's only 2 weeks. Okay, Drosophila melanogaster, it has very short lifespan. It has only 2 weeks. It consists of only 4 pairs of chromosomes. It consists of only 4 pairs of chromosomes. Okay, it consists of only 4 pairs of chromosomes. This Drosophila melanogaster, in single mating, it gives rise to many progeny. Progeny is nothing but the daughter individuals. Okay, progeny is nothing but the daughter individuals. In a single cross, this in a single mating, this Drosophila melanogaster, it will give many progeny. Many daughter individuals are produced. Okay, and this Drosophila melanogaster, it shows clear sexual dimorphism. It shows very clear sexual dimorphism. That means males and females are very clearly distinguishable. Males and females are very clearly distinguishable. Okay, so this Drosophila melanogaster shows a very clear sexual dimorphism. Okay, and Drosophila melanogaster has many different hereditary characters. Many different hereditary characters. Okay, many varieties of hereditary characters are seen in case of Drosophila melanogaster. Okay, so these are all the different reasons why Morgan has selected Drosophila melanogaster for his experiments. So T.H. Morgan is a person who wanted to prove experimentally the chromosomal theory of inheritance. For this reason, he has taken a fruit fly Drosophila melanogaster for performing his experiments, and it is having a very short lifespan, only 2 weeks, and only 4 pairs of chromosomes are present. Single mating will produce many progeny, shows clear sexual dimorphism, many hereditary characters are inherited. Okay. And the next thing about this Drosophila is one last thing, it can grow easily. It can grow easily on any medium. On any medium, it can grow very easily. Okay, these are the different features by which T.H. Morgan has selected fruit fly for his experiments. Let us now learn what experiments this person T.H. Morgan has performed. Okay. So, T.H. Morgan has selected a yellow bodied, white eyed fly. Okay, he took yellow bodied, white eyed fly and he crossed it with a brown bodied, red eyed fly. He crossed a yellow bodied white eye fly to a brown bodied red eye fly. Okay, so then he observed that the results, whatever the results were obtained, the results were deviated. The results were deviated from 9:3:3:1 ratio of Mendel. The results, as the same experiments like Mendel performed, right? Mendel has, he took in a dihybrid process, he took 2 different characters, like he has selected yellow seeds, yellow round seeds, he has taken green wrinkled seeds. In that way he has taken different characters together two different characters together. The same was done by Morgan. He took yellow bodied white eyed flies and crossed with brown bodied red eyed flies, but he observed that the first F1 generation's phenotypic F2 generation's phenotypic ratio was deviated from 9:3:3:1. This ratio was deviated completely from the Mendel's ratio. Okay, so then he identified that whatever the genes responsible for the color of the body and color of the eye, they were present only on the X chromosome. Okay, these two characters, okay, body color and eye color, these two genes are present on the same chromosome, that is on the X chromosome of the Drosophila. It was observed that these two characters, body color and eye color chromosomes, both were present on the X chromosome only. The genes for both are present on the X chromosome only. Okay. So, he also observed that there were more of parental combinations. Okay. In this cross, there were more of parental combinations are seen, more parental combinations were seen, and he also observed some non-parental combinations also. He also observed some non-parental combinations also. Okay. So, he observed some parental combinations and some non-parental combinations also. So, observation of the parental combinations and also non-parental combinations is showing us two things. Okay. The first thing is linkage. Linkage. This could be the reason, because linkage is nothing but the presence of two genes on the same chromosome, is called as linkage. The presence of two different genes on the same chromosome is called as linkage. Presence of two different genes on the same chromosome is called as linkage. The appearance of more parental combinations is due to linkage only. That means the physical linkage between the genes. The genes are actually present on the same chromosome in a linear manner. Okay, that is why these genes are inherited together. That is why we are able to see more of the parental combinations. Some non-parental combinations are also seen. Okay, this appearance of these non-parental combinations is due to recombinations. Appearance of non-parental combinations is due to recombinations. Recombinations is nothing but appearance of new combinations of genes, okay, which is due to lack of linkage. Okay, which is due to lack of linkage. No proper linkage is there. So, that is why recombinations were possible. Okay. So, students, there were linkage means the presence of two genes on the same chromosome is linkage. Okay. So, this linkage is actually almost all the genes present on one chromosome. Okay, all the genes present on one chromosome will form one linkage group. Okay, all the genes present on one chromosome will form one linkage group. Okay, like for example, as we have taken Drosophila is having 4 chromosomes, 4 pairs of chromosomes. Okay, so it is going to contain 4 linkage groups. Okay, it consists of 4 linkage groups. When we take man, in man there are 23 pairs of chromosomes. In man there are 23 pairs of chromosomes. So, totally in man there will be 23 linkage groups. In man there will be totally 23 linkage groups. Okay. In if we take a sweet pea, plant, there are 7 pairs of chromosomes. So, there will be 7 linkage groups. groups in case of sweet pea. Okay. In that way, how many pairs of chromosomes are there? That many linkage groups will be present. So, each chromosome will make up one linkage group. Okay. Then next we are going to learn types of linkages. Types of linkages. There are two types of linkages. The first one is complete linkage, and the second one is incomplete linkage. Okay, complete linkage and incomplete linkage. In case of complete linkage, the genes are located very close to each other. Okay, in case of complete linkage, the genes are very close to each other. So that all the time they are inherited together. They are inherited together, always inherited together. So, the offsprings they will be showing only parental combinations. Okay, the offsprings they will be showing only parental combinations. This is complete linkage. The genes are located very close to each other so that they are always inherited together, and the offsprings they will be always showing the parental combinations only. Okay. The next one is incomplete linkage. Okay. So, incomplete linkage means the genes are linked to each other but not as closely. Okay. The genes are linked to each other. The genes are linked to each other but not always inherited together. Okay. But not always inherited together. In this case of incomplete linkage, more than 50% of the offsprings show parental combinations. Okay. And the two different combinations of the two different parents are there, right? So, two different combinations will be seen. The two different new combinations will be seen by less than 25%. Because more than 50% is parental combinations only. So, two different new combinations will also appear, which will be like less than 25% each, because more than 50% is already over with parental combinations. So, less than 50 will be there. So, less than 25 will be shared by two different new combinations. Okay. So, this is linkage, and always linkage. Okay, the linkage is inversely proportional. Okay, linkage is inversely proportional to the distance. Okay, so the more the distance, lesser will be the linkage. Closer the distance, more will be the linkage. Less the distance, more will be the linkage. More the distance, less will be the linkage. Less distance, more linkage. More distance, less linkage. In that way, linkage and the strength of linkage and distance are inversely proportional to each other. The strength of linkage and the distance are inversely proportional to each other. Okay. If linkage is more, then distance is less. Okay. If linkage is less, that means distance is more. more. Okay. So, this is all about linkage. Then we have recombinations. Recombination. Recombination is nothing but new combinations from the parent type is nothing but recombinations. New combinations are of the offspring is due to recombination. Recombination is occurring due to crossing over during meiosis. Due to crossing over during meiosis, recombination is occurring. Due to crossing over during meiosis, recombinations are occurring. Recombinations are occurring because of crossing over during meiosis. Okay. So, here more is this recombination is dependent on linkage. Recombination is dependent on linkage. It is dependent on linkage. Okay. More linkage, the less will be the recombination. Okay. Less linkage, the more will be the recombination. Okay. For example, you see the yellow and white color of the fruit fly. Okay, this show only this yellow and white color body of the fruit fly, they show only recombination of 1.3 percentage. 1.3% recombination is only shown between yellow and white color fruit flies. And if you see white and miniature flies, if you observe, there will be 37.3% of recombination. Okay, there will be 37.3% of recombination. That means here yellow and white color flies are showing more linkage than white and miniature flies. Okay, that can be observed. So, students, here the frequency of recombination explains the distance between the genes, okay. The frequency of recombination explains the distance between the genes. The frequency of recombination, it will explain the distance between the genes, okay. So, students, by using this frequency of recombination, a student of Morgan called as Alfred Sturtevant, he established gene mapping. He identified gene mapping in which we will be using the frequency of recombination to map the distance between the genes. Okay, frequency of recombination is used to map the distance between the genes. Okay, so in this way, Alfred Sturtevant has invented gene mapping, in which by using the frequency of recombination we can understand the distance between the genes. And this gene mapping technique proposed by Alfred Sturtevant was very much helpful in the human genome sequencing. That means in identifying the sequence of genes in the human DNA, this was very helpful. Okay, it is very much helpful in identifying the sequence of genes in the DNA. Okay, so it was very helpful in identifying even the different parts of the human genome also. In that way, this gene mapping is very much helpful, okay, in which recombination frequencies are used. So, here students, this is all for today. In today's class, we have learnt about chromosomal theory of inheritance, we have learnt about linkage and crossing recombination. And one last thing students about recombination, what is the advantage of this recombination is, you know, new combinations of genes will be formed, and if they are beneficial, then they will be helpful for successful survival of the organism. New combinations of genes will be formed which may be beneficial for the successful survival of the daughter individuals. Okay, that is one thing, importance of this recombination. Okay, so that's all for today. In today's class we have learnt about chromosomal theory of inheritance, linkage and also recombination. Tomorrow we will continue with the next concepts. That's all for today students. Thank you all for joining the class today. We'll meet in the next class. Thank you all.